In the previous video, we talked about how to locate a gene of interest using the internet, basically the human genome browser, and we talked about how to use PCR to clone the DNA. Now we have to insert the many copies that we have created into a vector, something that will deliver it to the bacteria. So a plasmid is basically a piece of circular DNA that is normally found in bacteria. Right? So bacteria have their own sort of regular DNA, if you would, but in addition to that, they also have these circular DNA called plasmids. Plasmids kind of give bacteria special features. Okay, it might give them antibiotic resistance. It might allow them to grow little hair on them so that they stick to each other better and therefore more resistant to environmental changes. So as a way of exchanging genetic material, two bacteria can actually come together and they could exchange plasmids. Okay, and that's part of how bacteria can evolve. So because bacteria are so willing to take up plasmid, we can actually insert the DNA sequence that we've cloned with PCR into a plasmid, and then we can feed that plasmid to the bacteria. Let's take a look at something called restriction enzymes. These are bacterial enzymes that are used by the bacteria to cut DNA sequences, and they will cut it at a very specific location that are called recognition site. We'll talk about the recognition site in a bit, but the restriction enzymes are basically used by the bacteria to defend themselves against foreign DNA. So sometimes bacteria would pick up DNA from the environment, and they don't want those DNA sequences to alter them in any way. So the restriction enzymes are going to cut them out. The bacteria's own DNA will not be affected by the restriction enzyme because they are going to be methylated. Basically, what that means is, let's say there is a DNA sequence here in the bacteria, and what they will do is they will put CH3 on them, and this is called methylation. And by methylating them, it's going to protect them from the restriction enzymes. So the restriction enzymes will only cut foreign DNA that the bacteria may have accidentally picked up from the environment sometimes. So these recognition sites, these specific locations that are going to be recognized by the restriction enzymes, are usually 4 to 8 base pair long, and they are usually characterized by complementary palindromic sequence. A palindrome in English is basically a word that spells the same forward and backwards. Here are some examples: madam, level, wow, and kayak. So if you spell kayak this way or backwards, is the exact same word. You can even have a palindromic sentence. Was it a car or a cat I saw? If you read this backwards, it says the exact same thing. Was it a car or a cat I saw? Right, so that's an example of a palindromic sentence. So palindromes also exist within DNA, and here is an example of a palindromic sequence: GAA-TTC. The complementary strand is going to be GAA-TTC, the same thing, right? So forward and backwards, it's the same thing. So the restriction enzymes will recognize these palindromic sequences and they will cut them. Right? So here we have and restriction enzymes called ECOR-R1, which is basically restriction enzymes derived from E. coli. Okay, and it will recognize this palindromic sequence and cut it like this. Okay, so after you cut it like that, it will create two fragments with these we call it overhangs, okay, which means they are not going to have a complementary strand, and they just kind of hang out there. So here are more examples, a whole table of these restriction enzymes, and you can see some restriction enzymes like ECOR-R1 or SOL1 here or Hindi3 here, they all create these overhangs, okay? And if they cut and create overhangs, we actually call these sticky ends. because what happens is, since this overhang and this overhang are complementary, if you just kind of leave them in a solution for a bit, they actually will find each other, and then they will reanneal. They will come and form those hydrogen bonds again, and they will stick back to each other. So that's why they're called sticky ends. So here is another sticky end, another sticky end. But some of the restriction enzymes, they are going to cut straight through. Instead of creating like a zigzag cut, they will create blunt ends, and there's no sticky end here, and these are very difficult to reconnect in most cases. So for the purpose of inserting a gene into a vector, a plasmid, we would be using restriction enzymes that create these sticky ends. Okay, so equal R1, Sol1, and Hindi3 would be useful for the purpose of inserting a gene into a vector. So let's do a sample problem. We have a DNA sequence here, and we are going to be treating it with SMA1, and we want to know where the cuts are going to be happening and how many fragments you are able to generate. So something like this, right? We can look at the recognition site of SMA1, and it's going to look for GGGCCC from the 5' end to the 3' end. So we should scan from the 5' end on either side, and we look for GGGCCC. So if I go this way, right, we're going to look for GGG. So there it is, GGGCCC. So that's one of the recognition site, and when you expose this to SMA1, it will cut it right there. Okay, and we're going to look for another GGG. And there it is, GGGCCC, right? And we are therefore going to cut this as well, right? You could have looked for it from the other side, okay? If you go from the bottom strand, you will see GGG here, right? And you would know this is the recognition site as well. That's because, again, this is a palindrome, right? So it doesn't matter which way you go. Just have to make sure that you go in the same direction that is shown in the recognition sequence here, right? You cannot go from 3' to 5', right? If you do go from 3 to 5, then you have to be looking for CCC GGG, right, as opposed to GGG CCC in the other direction. So you know, how likely are you going to find these palindromic sequence in DNA? Well, you can do a little bit of math. Okay, if there are 6 nucleotides in the recognition site, then the probability of finding that would be 4 to the exponent 6. Okay, so if you plug that into the calculator, 4 to the exponent 6 is 4096. That means if the genetic sequence were completely random, the probability of finding something like this is once every 4096 base pair. Okay, so the longer the recognition site, the less chance that you're going to find it. So a few slides ago, we talked about some restriction enzymes creating sticky ends, other creating blunt end. Here is an actual example. We treated a DNA segment with some kind of restriction enzyme, creating the sticky ends here. And if I left them in the solution, then eventually this segment will find this, and then they will come back together. But it wouldn't be a permanent link. Okay, if you heat it up again, then you know it might separate again. Because if this segment finds that segment, they will just be held together by hydrogen bond. So for you to glue them back permanently, you have to add DNA ligase in the solution. So you can form the actual covalent bond between these nucleotides, and that would cause the DNA segment to come back together. In order for this to work, in order for you to glue these sticky ends together, it has to be sticky ends that are created by the same restrictions enzyme. So for example, EcoR1 creates sticky ends, and Hindi3 also creates sticky ends, but this sticky end will not be able to glue together with this sticky end because they are not complementary, right? So if you try to put them together, you can see we have a severe mismatch here, right? A doesn't go with A, and you know G doesn't go with A, so this will not be able to glue together even if you supply DNA ligase. So that means if we want to insert a DNA segment into a plasmid, we must treat the DNA and the plasmid with the same restriction enzyme. Here we have a plasmid that has a palindromic sequence and is going to be a recognition site for the restriction enzymes equal R1. We have to make sure that our gene of interest, the one that we're trying to insert into the plasmid, also contains the same palindromic sequence at the ends. And when you treat the DNA sequence and the plasmid with the same restriction enzyme, then you will be able to create sticky ends in the plasmid as well as the ends of the gene. And when you mix the two things together in a test tube or something like that, then this sticky end, I would call sticky end number 1, is going to connect with this part of the plasmid, right? Because AATT is going to be complementary to TTAA. Similarly, the other side, the TTAA here, we'll call it number 2, is going to connect with this part of the plasmid, the AATT, because they're complementary. And afterwards, if you add a little bit of DNA ligase, then you will be able to seal the gene into the plasmid, and we can then feed this into the bacteria. You might be wondering, well, how do you know that when you add the ligase that the plasmid won't just close up on its own without accepting the gene? And that's possible, and there are actually ways for us to tell whether the insertion occurred or not, and we're going to be talking about that in a later slide. So here is a typical plasmid. You can actually order plasmid from various companies now if you're doing an experiment. So a plasmid would come with a bunch of restrictions enzyme sites, and you want to choose a plasmid that contains the same site as the ones that's flanking your gene. Okay, so if I have a gene here I'm trying to insert, and the ends of the genes are going to have a restriction enzyme site for BAMH1, so you want to choose a vector that would have the same recognition site. So when you treat the plasmid and the DNA sequence with the BAMH1, they would create the same sticky ends that will allow the gene to be inserted. In the plasmid, there would also be the origin of replication. That's where the promoter is, if you would, and then the gene is going to be copied in this direction, and then there will also be antibiotic resistance genes in the plasmid. So here we have an ampicillin resistant gene, here we have a tetracycline resistant gene, and that's actually a way for you to verify later on to see if the bacteria has accepted the plasmid. So basically what that means is if you have a bacteria that does not have the plasmid, if you try to feed it the plasmid and it didn't take it, then what happens is when you try to grow it in a medium that has ampicillin, then it will die because the antibiotic will kill them. Whereas if you try to grow the bacteria in a medium that does not have ampicillin, then it's going to grow normally. Okay, so you can see this is what they call lawn colony. That just means they grow everywhere. If they accepted the plasmid that you feed them, because the plasmid contains the ampicillin resistant gene in it, then you would expect them to be able to grow. But not all bacteria is going to grow, okay? Because you will never have 100% transformation. You will not be able to make all the bacteria take up your plasmid. So only those that took up the plasmid will grow on the plate, and those are the ones that you are going to harvest and use it to mass produce whatever protein product is encoded by your gene. And if you grow this bacteria on a regular medium with no ampicillin, then there will be a lot of growth just like before, right? So let's go back to here. If you inserted the gene into this location instead of over here, then you would test the bacteria colony on something that contains the tetracycline, because if you insert a gene right here, then you actually break open the ampicillin resistant gene, right? So even if they accepted the plasmid, they wouldn't be able to grow in the presence of ampicillin because you disrupted the gene by inserting something over here, right? But if you insert it over here, you still leave the tetracycline resistant gene intact, and so that's why you're going to test it on a tetracycline culture instead of an ampicillin culture. If you insert the gene over here, then you can test it either on an ampicillin or tetracycline culture, because inserting a gene here will not disrupt the ampicillin resistant gene, and it will not disrupt the tetracycline resistant gene. So all this stuff, it's kind of complicated. I'm going to try to explain to you in a class note, and hopefully it'll make more sense afterwards. So let's start from the beginning. We first have to locate the gene of interest, and we can do that using the genome browser, and we found that the insulin gene is on chromosome 11. Right? So that's the location of the gene. We want to clone this gene, and we want to feed it to a bacteria so that it's going to make the insulin protein, which we then can purify it and give it to people with type 1 diabetes. So once you located the gene, we want to know the exact sequence of the gene, and so let me just draw the gene here. Here is the gene. It's going to be a double helix, and what we want to do is we want to use PCR to clone this. Okay, so PCR is a method that we could use to mass produce a specific copy of the gene, and in order for us to do that, we would have to design a primer. Okay, so there is a DNA primer, and the primer is going to be on the either ends of your of the gene of interest. Okay, so typically you want to have the primers that is a little bit upstream and downstream of your gene. So your actual gene might be from, say, here to here, right? That's like the insulin gene here, INS, and you want the primers to be a little bit upstream and downstream. Okay, so when you copy it, you will copy a little bit of extra stuff as well, and I'll tell you more about the extra stuff in a bit. But basically, what happens is after the PCR, you will be able to create many copies of this. Okay, so let's say you cloned it already. Okay, the next step you want to do is you want to feed it into a plasmid. You want to insert it into a plasmid. So here is a plasmid, and you can again buy these from various companies, and a plasmid would come with, say, a selective gene, right? A gene that allows you to choose the bacteria that has taken up the plasmid, and it usually gives the bacteria some kind of antibiotic resistance. Okay, so let's say we have ampicillin resistant gene right here. Okay, a plasmid can have multiple insertion site, but just to keep it simple, we are going to have an insertion site here for the restrictions enzyme equal R1, and the restriction recognition site for that restriction enzyme is GAA TTC. So it's a palindromic sequence, which means it's going to be the same when you read it backwards. Okay, so in order for us to use this plasmid, we have to make sure that in the flanking sequence that are around the gene of interest, there will also be the same recognition site. So over here, there has to be the same GAA, TTC, and CTT, AAG, and over here it will be the exact same thing. Okay, GAA, TTC, CTT, AAG. Okay, if this is a different palindromic sequence, then you would have to choose a different plasmid that match those sequences. What we want to do next is to treat this DNA clone with the equal R1, the restriction enzyme that will create the sticky ends, and from that, what you're going to get is you're going to get your DNA sequence like that. And after the restriction enzymes cut on both sides, you will now have some sticky ends. Okay, so the sticky end on the left side here is going to be CG, and then we will have TTAA. Okay, and this part is just going to be gone, right? And on the other side here, we would have the GC and TTAA at the bottom. Okay, so that's a sticky end and that's a sticky end. Then what you're going to do next is you will treat the plasmid with the same restriction enzyme. Okay, so you're going to do equal R1 again, and the equal R1 will cut the plasmid like so, because that's the recognition site, right? And so the plasmid is going to pop open. Let me try my best to draw this thing. Maybe it's easier if I just cheat a little bit, erase that, and then it's gonna pop open, and what you're gonna have is going to be a G here with a C TTAA, and over here, let me pop it open a little bit more. We are going to have the other end, right? We're gonna have the C TTAA, and at the bottom it would just be, oops, sorry, CTTAA, and at the bottom here would just be a G. Okay, so now if we mix the DNA sequence and the plasmid together, then what you will see is this part here is going to anneal with this part, right? Because you can see the AATT is complementary to the TTAA, right? And similarly, this part over here is going to anneal with this part, okay? Because again, they are complementary. So if all goes well, right? After you mix them and you add a little bit of, so we'll call this A and we'll call this B. So if you take A plus B, mix it together, A plus B, and you add some DNA ligase, okay, then what's going to happen is you're going to end up having a plasmid. There's your plasmid with the gene of interest inserted in it. Okay, so that's the gene, right? That is the INS gene, the insulin gene, and don't forget over here you are going to have the ampicillin resistant gene. Okay, it was always there, right? So there is the AMP resistant gene. Okay, so now you will feed this plasmid to say a E. coli bacteria. Okay, so you would have a solution, a vial that contains E. coli, okay, the bacteria, and then you're gonna add these plasmid to the mixture, and there are actually several ways you can encourage the E. coli to take up the plasmid. We are not gonna go into details about what those methods are. So after you let the bacteria take up the plasmid, how do you verify that? How do you know the bacteria has actually taken up the plasmid? So you will have to take this tube of bacteria and you would have to grow it on different cultures. Okay, so you're gonna have a petri dish, and this petri dish is going to have just normal medium. Okay, so the medium is like with the nutrients that's necessary for the bacteria to grow. So if you put the E. coli on here, they should grow. Okay, if they did not grow, that means they somehow died in the process of the transformation. Okay, then you are going to put them in a medium with antibiotics. Because the plasmid contains a ampicillin resistant gene, we are going to add ampicillin to it. Okay, so when you put the bacteria in the medium here with no antibiotics, and you put it in the medium with antibiotics, what you expect to see is there is going to be a lot of bacterial growth in the first one right here. Okay, so you're gonna have a lot of bacteria growing. These are bacteria. Okay, they're growing all over the place because it's just nutrients in the plate, right? Why wouldn't they grow? Over here though, because you have ampicillin, then you will see isolated colonies. Okay, so only bacteria that has taken up the vectors would be able to survive in this environment. Okay, remember, you are not going to be able to make all the bacteria take up the plasmid. Okay, only a percentage of the bacteria will be able to do it. So in this solution, only the bacteria with the plasmid containing the resistant gene is going to be able to survive in this medium. If it's just a regular bacteria that did not take up the plasmid, then it will not have the antibiotic resistant gene in it, and so it's not going to be able to grow. Okay, so by putting it into these medium, we are able to select for the colonies that has taken up the plasmid. So what you can do is you can isolate these colonies, okay, and then you can put them in another growth medium, but this time like a much bigger vessel, for example, like a whole flask, and then you just let the bacteria grow in there, and what they will do is they will also be producing the insulin, right? Because the plasmid that you have given them also contains the insulin gene, right? So after a while, you will have insulin in the solution, and you'll be able to extract them and purify them, and that's how you create the insulin in a large scale. Okay, so this is an example of how to use recombinant DNA because you're inserting a human gene into a bacterial plasmid, and recombinant DNA is basically a DNA sequence that contains DNA from two different origins, right? And you are able to use this technology to mass produce insulin. So I hope that kind of makes sense. I'll try to find a link that has a nicer animation than these drawings. And post it on eCentennial, and probably that will help you further understand the process a little bit more.